Big breakthroughs take time. Long before any cures are found, decades of research are often needed to simply understand a disease. Now, more than any other point in history, researchers are poised to make major breakthroughs in genomic medicine.
The more we learn, the less we know. The horizon is forever receding, but nevertheless, the information we do gather can be very helpful. And while I see many tragedies, I also see amazing success stories.
Genomics is a single word, but it encompasses so many aspects of what medicine is doing to try and help people.
That genome, which is one genome, is hardwired, is being used in countless different ways, all through development, all through life, and in every moment that we're exposed to our environment.
But when you think about medicine generally, like, conquering diabetes, the development of early vaccines for polio, you never go from your first swing at the bat to a home run. You hit a lot of singles and doubles. And for a lot of genetic diseases right now, I think we're getting a pretty good batting average, but we're not power hitters yet.
Today, gene editing technology is helping advance discoveries in medicine, discoveries out of reach just 5 years ago. These tools are helping unlock new paths for research and, ultimately, cures for disease.
In addition to the revolution in stem cell technology, there's also been tremendous advances in the last couple of years in genome editing. So the ability to, first of all, just sequence all of our genomes and understand where potential mutations might be, but also using new technologies called CRISPR Cas9.
CRISPR Cas9 technology allows researchers to quickly and accurately produce genetically modified mice. These mice can then be used to learn how genetic diseases affect the body and how these genes react to new types of medicine.
The nice thing about CRISPR is it's so easy. Now we understand the CRISPR more. So there's a new version of the CRISPR coming out constantly. So I'm sure that the tool will be getting better and better in the future, and to the point like that we can use it safely in patients.
It's all new all the time. Every week we're doing something different. So it's cool because I feel like I get to learn a lot of new techniques, and we're just always coming up with a new approach to, a different problem.
Driven by the CRISPR revolution, this lab's work in genetic engineering will positively affect the future health of children around the world.
Probably cystic fibrosis is the best example. There are more than perhaps 2,000 mutations in one gene. Simply changing a base pair on a child's cell that would totally restore his cystic fibrosis, turning him into a child who doesn't have to go to the hospital 3 times a year, would be a godsend. It would be a wonderful thing for that family and that patient. So this gives us an optimism that we never could have conceived of 20 years ago.
Now, this gene editing technology is helping scientists understand and treat disease in human organs in ways we could only imagine a few decades ago.
Here at the Children's Hospital, unfortunately, we see a lot of patients that come through with congenital defects of organ development. And in order to understand the genetics behind how that occurs, we need to understand how organs form in the first place.
So I think one of the most exciting early discovery projects here is our organoid center. We call them organs in a dish because they're very small, maybe 1 to about 10 millimeters in size, but they do everything.
Starting with tissue in a lab dish, scientists are learning how to coax small numbers of human stem cells into forming complex, 3-dimensional tissues that mimic the functions of entire organs.
Each of these are about a couple millimeters in diameter, and this is human intestinal tissue that's been grown from stem cells. And this takes about a month to grow these. They didn't set out to try to generate these organoids. They were trying to just see if they could make human intestinal tissue in sort of a layer on the dish, and then those cells just innately tried to self-organize and made these little spheroids and organoids, and that's the thing that just blows your mind.
To cure that child by genetically engineering from a single cell to form a liver, or a kidney, or a stomach, or an intestine. And now that's within our grasp. So, You wait 40 years for these new horizons, and sometimes you're very fortunate to be able to see them come into view in the span of just one career.
Today, growing organoids in a dish can help scientists safely test new drugs without having to rely as much on animals. Someday, this groundbreaking work may also allow researchers to grow full-sized replacement tissues that could reduce or even eliminate the need for organ transplant waiting lists.
I think it's still important to keep an eye on the long-term view, and that the basic research that we might not understand what the use is immediately, in the long term, some of the serendipitous discoveries are the most important ones. Discovery is a bit of a high, and you just, from one discovery to the next, one discovery leads to another question, and it's ultimately just trying to follow that line of questioning.
The future we can predict will be filled with medical discoveries that will not only teach us about who we are as individuals, but also what we all have in common within humanity's code.
It is humanity's code, but it's a code that fortunately we don't understand that well yet, and probably we never will. So we'll always have the mystery with us of what it means to be human. And I think of the code as reminding us both of what we know and what we don't know, and bringing us together around that.